Good morning, students. Welcome to webinar sessions of Model School for Junior Inter. Children, today we have Zoology, English, Mathematics, and Political Science sessions. In the previous sessions of Zoology, Miss JHANSI ma'am explained us about connective tissue, isn't it? Do you remember how it was categorized? Yes, it was categorized into proper connective tissue as well as specialized connective tissue. Proper, madam explained us about that proper can be categorized again into loose connective tissue as well as dense connective tissue, isn't it? So what is there in the loose? In again, it is categorized as areolar, adipose, and reticular, isn't it? And in dense, what we have discussed, we discussed about the regular, irregular, and elastic. So we have totally, ma'am has totally completed the proper connective tissue. Now she started about the specialized category of connective tissue. In that, ma'am clearly explained us with examples about the cartilage, isn't it? Actually, it is divided into cartilage, bone, and blood, isn't it? So ma'am completed cartilage too, isn't it? Elastic, hyaline, as well as fibrocartilage. Now let us learn detailedly about bone as well as blood connective tissue. Okay, children. So I welcome JHANSI madam. JHANSI ma'am is from Munipalli Mandal as well as Sangareddy district. Okay. Good morning, madam.
Ma'am, good morning. Ma'am, thank you very much. In brief, I should say that you have summarized whatever we discussed yesterday with the students. Hope that yes, they have gone through the textbook. Just one point I missed out yesterday when I was discussing adipose tissue. Dear students, you might have found that the skin that is present over here in our palms and soles, you find the skin over there very thick. It is because of the presence of adipocyte tissue, adipocyte cells. And this tissue over here actually provides the cushioning type, or they work as shock absorbers when we try to hold some object which comes with greater force. So there, this is helpful. That is why this part of the skin is different. Now today we are going to continue with the specialized tissue with the hello. Today we are going to continue with the bone and fluid connective tissue. So let's get started. Ma'am, can you see the screen?
Yes, I can.
Yeah. Okay. Okay. This is all revision of yesterday what we have seen. We finished all this. So this is specialized connective tissue. So we stopped here with bone, right? Let's get started with this tissue. Bone, osseous tissue, it is also called as. Study of bones is called as osteology. In fact, you might have seen people visiting orthopedics who deal with bone problems. Whenever there is fracture or there is a problem with bone where the bones become very weak, brittle. So how do we define bone? Bone is a solid, hard, mineralized, calcified tissue, connective tissue. It is solid. It is hard, very hard. Why it becomes hard? Because of the process of mineralization. When you use the word mineralization, we are talking about here deposition of minerals. What are the minerals that make up bone? Calcium and phosphate, magnesium, these are the salts, mineral salts which are important, which makes the material very hard. The bone becomes very hard because of this. So it is a calcified, mineralized, solid connective tissue. One difference between bone and cartilage is that cartilage is pliable. You remember I showed you a picture of a rubber band, rubber strip trying to bend it. So bone is not pliable. If bone were to be pliable, then our body would have been very flexible, totally. There wouldn't have been any need for bones only, because cartilage would have done the work, as in the case of chondrichthyus fishes. But here bone is non-pliable by nature. If at all bone becomes pliable, then you go to hospital with nothing but fractures. So bone is said to be non-pliable in nature. It is made up of calcium, magnesium, and phosphate salts especially. So important part of bone. How many bones are there in our body? There are totally 206 bones present in our body. This is all in short the bones that make up our body. So this is the femur bone or the thigh bone. which is the longest bone of our body, and the smallest three bones are present in our internal ear. Maybe yesterday you remember I showed you eustachian tube. So there in the middle part of the ear there are three bones: malleus, incus, and stapes. Stapes is the smallest bone of our body. So there are totally 206 bones in our body. So before getting into the details inside the tissue of bone, let's try to understand the other part. So this picture represents that yes, it is a mineralized part. That means minerals become hard, deposition of especially calcium carbonate, calcium phosphate, etc. Now the other functions of bone. When you look at the skeleton, the bones is nothing but we are talking about skeletal system inside. Because the bones are present inside the body, we call this as endoskeleton. Endoskeleton. Anything which tries to give you shape, protection, everything from outside, then we call it as exoskeleton, as you find it in case of cockroaches. Cockroach get its shape, protection from outside layer, which is exoskeleton, which is nothing but again it is a cuticle which helps it out. In our case, our skeleton is inside the body, hence it is called as endoskeleton. So bones, when you look at the bones, the way they are arranged, you can easily try to guess the animal. That means our shape, bones work as a framework for our body, giving shape to our body, support to our body. Now, we already studied about skeletal muscles. You remember when I discussed muscular tissue. So muscles and bones, they work together and they help us in locomotion, in movement. So we call this as together, we study together actually under the heading musculoskeletal system. So skeletal muscles are attached to bones. Skeletal muscles are attached to bones which help in locomotion and movement. And we also learned how the skeletal muscles are attached to bones. It is through the tendons. So these are the different types of movements, whatever you do in your daily life, right from morning till evening, walking, jumping, curling, etc. And if you're standing in one place and moving your body parts, like for example, right now I'm sitting over here and moving my hands, showing gestures. So this is movement. If I shift my position, my body's position from A to B, then it is movement. Sorry, locomotion. So locomotion and movement takes place because of bones and muscles working together. Another function of bones help and they try to protect the inner delicate organs. So you can very easily understand looking at this picture that these two, here we have a pair of lungs which are well protected in this bony cage. And you remember yesterday I showed you the sternum breastbone, and these are all the rib bones. Rib bones are attached to the sternum bones. Over here, yesterday picture I showed you hyaline cartilage, right? Sternal costal cartilages. See, I'm trying to even connect with what we have learned yesterday, if you can connect it. So these are the ribs which are present over here, backside. This is our backbone. This is our backbone, right? So these are the ribs. So inside, delicate organs are protected. That is our lungs and as well as heart, which is present in between the lungs. Bones also bear the weight of the body of the organism. And this is the reason why you find fat people who are overweight or obese people, they suffer from knee problems because their entire body's weight falls on the knees. That means actually the limb bones, they take up the weight of the body. Even if you consider very largest animals moving on the earth, like elephants, tallest animal like giraffe, the entire weight is taken by limbs. Now, let's try to look at bone. How does, I have taken over here, for example, our limb bones. Yesterday, I showed you even this picture. So this is a limb bone, how it appears. The free surface of the bone over here, this is called as epiphysis. Epiphysis free surface is again made up of articular cartilage, which is hyaline cartilage. Even this side, this is epiphysis. In between, there is a white line. You can see epiphyseal plate. This is also made up of hyaline cartilage. Then in between, very small region is there, which is called metaphysis. Metaphysis is the growing region of the bone. It elongates, it grows. Only then the bone grows. And then this shaft, rod-like structure which is present from metaphysis to this metaphysis beginning, in between this long stretch-like structure, rod-like structure is nothing but diaphysis. So the same thing over here, I have pasted another picture. This is to make you understand that inside the bone, bones are not hollow by nature in our mammals group. In our group, bones are filled up with air and they are called as pneumatic bones, and this takes place in birds. In birds, you find the bones in between, there is a hollow, this is a pipe-like structure, there is hollow space present which are filled up with air. So they are pneumatic bones. But in our case, the space that is present inside the bone, we call it as a marrow cavity, and it is filled up with material called as bone marrow. So to show you that, I have used this particular picture over here. So again, you have this is long bone because I have taken limb bone. So this is a free surface, and the long shaft diaphysis, which is filled up with bone marrow. The cells that make up bone. See, when we are talking about connective tissue, if you remember yesterday, my first slide when I discussed connective tissue, I said that connective tissue consists of the cells, and these are the cells which secrete two things, fibers as well as matrix. Matrix is very important when we talk about cartilage and when we talk about bones. So the bones cells which secrete this matrix of the bone cells, we call them as osteocytes. The first cell which is released from bone marrow, from our bone cells, bone marrow, we call this as osteogenic cell. That means it is called progenitor cell. It will give rise to, see, remember one thing, when we are talking about RBCs, WBCs, directly they don't come from the body, from marrow cells, bone marrow cells, fully fledged RBCs, fully fledged WBCs. It doesn't happen like that. First, a progenitor cell is prepared, it is produced. This undergoes process of maturation, development, and then it becomes functional. So like that, first bone which have not become functional, it has not developed. So that bone cell is called as osteogenic cell. This osteogenic cell, slowly it develops and it becomes into osteoblast. You remember, I also taught you the difference between osteocyte and osteoblast. When you use the word blast, it means it secretes, in case of bone, it secretes matrix. If this osteoblast doesn't secrete matrix, that means it is in inactive form, then we call this as osteocyte. So osteocyte have, over here you can see there are processes, branches like structures, which we call it as processes. These are called as canaliculi. They are called as canaliculi. This is meant actually to form a network or connection with the other cells. So we can find canaliculi. And then we have the final stage of the bone, which we call it as the osteoclast. So let's try to look at this particular structure over here. Again, in short, we can understand how a bone looks like. First thing is that you have matrix all around. Matrix is present all around. Right, matrix is very important. Then this is bone cell, and here are these all processes which we call it as canaliculi. And I also taught you yesterday in the introductory class about the spaces. In the matrix, you find spaces present. Inside those spaces, these cells are present which secrete matrix or fibers. So those spaces are called as lacuna. So here also you can see there is lacuna present inside. So matrix, lacuna, inside lacuna you have cells. So bone cells are called as osteocytes. We call these as mature cells. Then these osteoblasts are the cells which secrete the matrix around it. And this osteoblast comes from the cell called as osteogenic. That means this develops into this. And finally, the last stage of this bone cell is called as osteoclast. Osteoclast are worn out cells. That means you find these kind of cells, especially in aging people. So these are worn out cells. They help in the process of something called as resorption. And these also take up the function of phagocytosis. That is, they try to engulf bacteria or any other germ particles. So they do the work of even phagocytic cells. Another important point about osteoclast, as I mentioned, that they help in resorption of calcium. There is connection between blood and bone. So what is the connection over here is that from blood, whatever calcium is needed for the growth of matrix, for the well-being of the matrix, that calcium moves from blood into bone. It absorbs. But whenever you don't take sufficient amount of calcium in your body that is needed, that time, whenever the calcium levels fall in your blood, from bone, the calcium is taken back into the blood. Right? That takes place because of these cells which are osteoclasts. They help in resorption of this calcium. Actually, bone is the reservoir of the salts. Calcium, magnesium, phosphate, carbonate, salts, phosphates, everything you find over here. So under the influence of hormones, you find the movement of the material from blood, because whatever you eat, it is actually going to blood. Blood is the one which transports everything. So from blood, it reaches bones. Whenever your blood needs it, that is taken back. That time your bones become weak actually. That is the reason, see, we have to take balanced diet to see that all the organs function properly in our body. Okay, next, types of bones based on the method of formation. So which part actually becomes ossified or gets ossified and becomes hard to become a bone? Let's see that. Based on that, we divide it into 4 important categories. And the first one is cartilage bones. Cartilage bones, as you can see in the picture itself, that these are all the bones, limb bones, your forelimb bones, the hand bones, your hind limb bones, leg bones, and as well as both girdles to which these limb bones are attached. So this is our pectoral girdle over here, and this too we have studied yesterday, pelvic girdle, few of the bones over here. So these bones are derived from cartilage. That means when we are in the growing stage, that is fetus, embryo, or immediately after birth. So these bones are not directly hard bones in our body. Suppose you catch hold of young babies, infants, months old babies. Do you find in their body hard bones as you find in ourselves? No, it is not, because they have this cartilage in their hands. Cartilage is present. Slowly, as they keep growing, the cartilage material gets ossified. It becomes hard and gets replaced with bones. So this is called as cartilage bones. Another example for cartilage bones, we call it as replacing bones or endochondral bones. Endo, inside, chondral, cartilage. So endochondral bones. Next, another one is investing bones. The bones that are derived from the mesenchymal cells, embryonic mesenchymal cells, those are called as investing bones. Other two terms for this investing bones is membrane bound bones. They are called as membrane bound bones or dermal bones. Dermal bones. So examples for these investing bones, they are the 8 bones of our cranium. Cranium is a bony box inside which our brain is located. It is well protected. So there are 8 bones which make up the skull of our body. So this skull consists of temporal bones, occipital bones, frontal bones, parietal bones, all these bones. So they are derived from the cells called as mesenchymal cells, embryonic mesenchymal cells. What is the meaning of this embryonic mesenchymal cells? Just now I told you that beginning you find some kind of cell which is not differentiated, and later on it gets differentiated, matured, and then it becomes and does that important function. So to explain this particular slide, which is related to this particular investing bones, I'll come back in future again further now. So for the moment, remember that investing bones, they are derived from embryonic mesenchymal cells. Mesenchymal cells are mesodermal cells that are present. They become hard to give rise to these bones. So I'll come back to this because it has got connection. Sesamoid bones. Ossification of tendons. That means some places tendons become hard and get converted into bone. What is this tendon? Just now I told you, and even yesterday I shared with you, what are tendons? Tendons are the connective tissue connecting dense regular connective tissue which connects bones with muscles. So some of the tendons become very hard. So one such example over here is this pisiform bone, which is found in the wrist of our mammals over here. This is one example for this, and the second example is our knee bone, kneecap bone here, patella. This is called as. So these 2 bones are the examples of sesamoid bones. Now another last type of bone from where it is formed is visceral bone. So what is viscera? Viscera means we are talking about here important soft tissues and internal organs. So some of the soft tissues become bone. They become hard. So there are 2 examples for that. One example is over here. You know what is this? This structure is called as penis, which is required it is an intromittent organ required to transfer sperms into the female's body. So some of the ruminants, you find there is the soft tissues over here which is present. This becomes hard and it becomes bone, which is called as os cordis, os penis. Similarly, in the heart of ruminants, you know what are ruminants? Grass-eating mammals. And not all grass-eating mammals. Actually, when you use the word ruminants, we are talking about the animals like cows, buffaloes, which chew the material, whatever they get, and swallow it. Later on at leisure, they bring back the material into their mouth and start chewing it properly, thoroughly. That process is called rumination, and the animals are called ruminants. So in some of the ruminants, the heart over there, in the heart, they find one soft tissue which is called as os cordis. That becomes very hard, and it gets converted into bone. So some of the soft tissues get converted into bones. let's try to understand inside the bone. Now we understood from outside how bone is divided into three parts. Lengthwise we have seen epiphysis, metaphysis and diaphysis, etc., both the ends. So this is outside layer. Outside layer is called as periosteum, the outermost layer, and then this is the bone marrow cavity. The space is cavity and this is bone marrow. And this, can you see as I'm moving this cursor, this is the endosteum. That endosteum touches the inner hollow space where this bone marrow is connected. That means bone marrow comes in contact with the marrow spacing, with the marrow lining surface. So same thing I have placed over here, a vertical picture. So here once again you can see the blue line. Let's go from outside. The blue line that you can see here is periosteum, the outermost layer, right? Outermost layer of bone, periosteum. And then you find this is the inner surface lining. I'm saying what I'm talking about. The bone is endosteum. This is endosteum. Endosteum is in touch with this bone marrow. This space is called cavity, marrow cavity, and this is called as bone marrow, yellow color, what you are able to see over here. So from here to here, whatever is the structure present, we are going to study that under in detail. And that is very important for your, from your examination point of view also. So from here, the outermost layer, blue color, to this innermost, lining this yellow color material. This is the main TS that we are going to study now in detail, very important. So this is the transverse section of bone. This is the marrow cavity touching that yellow color what I showed you earlier. So here you can see that outermost is periosteum. Periosteum and endosteum both are made up of fibrous connective tissue. Both are made up of fibrous connective tissue. All this material what you are able to see everywhere around, all this what you are able to see, right? The white material that you can see over here, in fact white color, all that is matrix made up of the inorganic material, that is all calcium salts, etc. Now in between this matrix, right from periosteum, right from this periosteum till endosteum, you can find there are certain star-like structures present all around it. So these star-like structures over here, very close to periosteum and very close to endosteum, both the ends, they are present in circumferential pattern. So they are called as this very close to periosteum. We call this as outer. One second, my cursor. Yeah. So this, sorry for the technical delay. So this is outer circumferential lamellae. This side we call them as the matrix, and this black colored stars, we call this as, don't label it as black colored star. I'm going to teach you what it is. So this is outer circumferential lamellae. In the same way, very close to this endosteum which is present over here, very close to this also you have that longitudinal circumferentially placed lamellae, outer circumferential lamellae and inner circumferential lamellae. Now in between also you find the matter present, but in concentric rings. So this is concentric lamellae over here, lamella singular, lamellae over here. So in concentric rings it is present. Now what are all this? Now let's study about this black color stars. These black color stars are nothing but they are the spaces present, which we call it as lacuna, and inside that you find osteocyte cells, what I taught you in the beginning, osteocyte, osteoblast. So cells of the bones are present in this black colored material that you are able to see. So that black colored material is nothing but lacuna. Inside that lacuna, bone cells are present, which are our osteocytes, osteoblasts, osteoclasts, those are present. And the function of that one is to secrete the white color material around it, which is nothing but matrix. And that matrix is made up of calcium and phosphate and magnesium salts, phosphates, bicarbonates, etc. Right? So you have understood that. Now let's come to this important another point over here, which is called as Haversian canal. Dear students, there is one important difference, rather second important difference between cartilage and bone. So first difference is that cartilage is slightly pliable, you can bend it, but bone is very hard, you can't bend it. This is one of the important differences. Another one is that in cartilage we have learned that it is avascular, it is not supplied with blood vessels, but here bone is supplied with blood vessels. Bone is supplied with blood vessels, it is vascular in nature. So the point where the supply of the blood takes place, wherever you're talking about supply of the blood, you're talking about blood being carried in a vessel called as artery, blood being taken back to heart in another vessel which is called as vein. So wherever these two are present, automatically there is another capillary present or blood vessel present, which we call it as lymphatic vessel. So these three together exist, and along with, of course, the nerves. So here, this Haversian system, what you're able to see, Haversian canal we can call it, or Haversian system we can call it. This consists of artery, vein, and lymphatic vessel. These three are present over here, like over here. So surrounding this, this is one Haversian canal, this is one Haversian canal. So like this, you find all Haversian canals. All the Haversian canals are placed parallel to this marrow cavity. They all occur parallel to marrow cavity, bone marrow. So around this Haversian canal, in concentric rings, you find these bone cells present. And in between, whatever material is present, between this concentric rings and this concentric rings, in between, here and there, leftover spaces, like this is leftover space. So we call this as interstitial space. That is also filled up with matrix. That is also filled up with lacuna, with bone cells, and that is called as interstitial lamellae. This is interstitial lamellae. So the things which don't find, which are not exactly in the concentric rings and surrounding the other rings. So in between whatever lamella is present, that is interstitial. So what we understood, let's go over this once again. So TS of bone, I'm coming from outside. That means in the previous we have studied, right? Periosteum. That is from outside we are going inside the bone. So outside is blue color material. That blue color material over here is our periosteum. So slowly when you start moving inside, you find there is matrix over here, cells over here, and then endosteum in touch with bone marrow. That cavity filled up with bone marrow. So between the periosteum and endosteum, you find matrix present. The matrix that is present over here, very close to periosteum, we call that one as a circumferential lamellae. This is outside. And in the same way, you find over here inside circumferential lamellae, right? And then parallel to this bone marrow, you find spaced everywhere around Haversian canal or Haversian system. So what is present in Haversian canal or Haversian system? You find actually bone is vascular in nature. That is, it is supplied with blood vessels. So this has artery, vein, and lymphatic vessel. Surrounding this, you find all matrix made up of calcium salts, phosphate salts, etc. And then you find this material which is black color in nature. Here black color I have shown, but those are the spaces which are called lacuna. Inside lacuna, bone cells are present, and these bone cells secrete that matrix that is needed. Along with this matrix, another important point of bone is that it has got collagen fibers. Fiber is also secreted by these cells. Now, in the previous cell, I showed you that these cells have got the processes, right? These processes. So all these cells are interconnected with each other through the processes. They are tightly bound. They are also connected with this Haversian canal. And these Haversian canals are connected with bone marrow on one side and with periosteum on the other side through one canal, which we call them as Volkmann canal, Volkmanns, Volkmann's canal. They are connected with this Volkmann's canal. So I hope that you understood structure of bone and tiers of bone. Now, one Haversian canal is also called as one osteon. So you people have learned in 10th class that for nervous system, neurons are the structural and functional unit, right? In the same way for kidneys, nephrons are the structural and functional unit. Like that, for bones, we call Haversian canal or osteon as a structural and functional unit. So just look at this. This is the Haversian canal that we talked about, and this has got over here an artery which is bringing the blood from heart, supplying nutrients, gases, etc. We have vein which takes away all the material away, and wherever these three are present, I also told you, you have nerves. Always remember these four exist together: artery, vein, nerve, and lymphatic vessel. So these four are present in this Haversian canal. Haversian canal is surrounded by matrix. These are lacuna. Lacuna is white space. Blue color over here is nothing but osteocyte cells. So one Haversian canal with the surrounding matrix, with the surrounding lacuna, with the surrounding lamellae. We call this as osteon. This is another important question for your exam. Okay, again, we find the bones being divided into two types. One is called as a spongy bone and the other one is called as compact bone. We have already seen what the spongy and contact bone is, but we did not understand earlier. So when you look at limb bone, as I showed you in the earlier pictures, so the last part of the bone, the free end of the bone, which we call it as epiphysis over here. This epiphysis bone is present over here just like sponge. You know what is a sponge that we use sometimes for rubbing, wiping the material on the board, or even at homes, right? Even for plastering walls, etc. Sponge. So you find sponge being filled up with matter, and then there are holes, space present everywhere around, pores. In the same way, here also in spongy bone, entire bone is present in the form of actually irregular columns, which we call it as trabeculae. It is present in the form of columns, which is called as trabeculae. This can be trabeculae, we can call it as even cancellous bone. Cancellous bone. Now, whatever spaces are present over here, interstitial spaces, what we call it as, in all these spaces, you find blood vessels as well as bone marrow. So this is spongy part of bone. It is not regular. It is not completely compact, hard in one place. It is being spread out like network. So this part of the bone is called spongy bone. Appearance is like sponge. You can see over here, same, what I mentioned earlier, spongy bone. This part is called as spongy bone, right? So the rest part of the bone from here to here, the diaphysis of the bone is nothing but it is hard bone. That is only compact bone, right? So this hard bone, diaphysis part from here till over here, right? Diaphysis of the bone is compact, whatever we have learned earlier. So bone existing in the form of a spongy appearance. This is spongy bone. The bone over here is actually in the form of irregular spaces, which we call it as trabeculae. With that, we come to the end of bone, one of the specialized tissues we finished. So bone and cartilage. Now let's move on to fluid connective tissue. What is this fluid connective tissue over here? Fluid, you know the meaning, you have already learned. It is something which moves from one point to another. So our liquids and gases are considered as fluids. So blood is fluid connective tissue here. Along with blood, we also have another material, which is again connective tissue, fluid connective tissue, a part coming out from blood, which is nothing but lymph. Remember, students, even in 10th class, you have studied that you have two transport systems present in our body. One transport system is the main circulatory system that you have studied. Another one is parallel to this venous system, which is present, or accessory circulatory system, which is present, which is nothing but lymphatic system. So these are two systems present. So let's get started with the fluid connective tissue. One difference between fluid connective tissue, blood as connective tissue, and the other connective tissues that we have learned is that there are no fibers in blood. There are no fibers in blood. Blood doesn't have any fiber. It is fluid. It doesn't have fibers. It has matrix and it has cells. So in short, when you look at this picture, you can understand everything about blood. So blood, first thing is that blood moves through the vascular vessels, which we call it as blood vessels, which are arteries, veins, and connecting these two are capillaries. So blood moves through that. It is an important part of circulatory system, as I mentioned to you earlier. So blood consists of 55% of plasma. The liquid part, this plasma, the liquid part of the blood is only matrix. It is matrix for blood. And then you have the other cells present in blood. And these cells are divided into different categories. You already know them: RBCs, WBCs, and platelets. So these are the different cells that are present in blood. Let's talk about the other functions of blood. Blood helps us in carrying oxygen, in transport of gases, in transport of nutrients, and all those things we know, right? So on an average, adults have 4 to 5 liters of blood, and study of blood is called as hematology. So let's start first with matrix part of blood. Matrix part of blood is nothing but it is plasma. Now this plasma is, what is this plasma made up of? It has 92% of water. It is nothing but water, what we drink. And then it has got solutes which get dissolved in it. Plus it has got gases. So just now I told you, blood is the one which carries oxygen from lungs to tissues. picks up carbon dioxide from tissues and takes it back to lungs via heart, of course. So plasma is the one which carries over here. Of course, we have another important pigment also which helps in carrying oxygen. We'll come across it again. So let's look at this dissolved solutes. So what are the solutes that are dissolved over here? Organic substances which are present over here. These are plasma proteins. What are the plasma proteins? Alpha, globulins, heparins, all those things. The other material which is present over here, which constitutes 9.1% of the organic substances, they are lipids, enzymes. Lipids are less in number, of course, because lipids, fats are specially taken up, carried by lymphatic system. Enzymes, which is secreted. Hormones, which keep moving from the place where they are produced to the target organs. You remember when we were doing epithelial tissue, I told you about the tissues being divided into ductless tissue glands and without ducts, with ducts and without ducts. So glands which don't have ducts, they secrete their secretion, which we call hormone, into blood. So it is a plasma which carries that. So plasma carries enzymes, hormones, nutrients, waste. What are the waste products over here? Of course, we are talking about nitrogenous waste products. So urea, uric acid, hippuric acid, creatinine, all the nitrogenous waste. All this is carried by blood. We know that blood is one of the most important system of transport system, circulatory system. So it carries. So who carries in all these things? It is a plasma which takes place. It carries food. Whatever food you eat is digested in the intestine and finally given it to blood. So blood carries that amino acids, glucose, all the things that is needed over here. So this is actually the summary of all that, what plasma does. Now, plasma proteins. What are the plasma proteins? As I mentioned earlier, albumin, globulins, fibrinogen. These are the plasma proteins. Apart from this, there's one more missing out in this, and that is heparin. So these are fibrinogen is produced in liver. Prothrombin, there's another plasma protein which is not over here in the slide. Prothrombin, fibrinogen, heparin. These three are produced in liver and then secreted here in blood. So these are the plasma proteins. We'll go through the functions of albumin and globulins, which are very important. Again, these are the different salts that you can find here in plasma. Plasma consists of sodium, potassium, calcium, magnesium. Very important, these salts. In what forms? Chlorides, sulfates, phosphates, bicarbonates, all these salts are present in plasma. Gases, I already told you, oxygen, carbon dioxide, nitrogenous waste material, regulatory substances, all these are the things carried. So plasma proteins, which are missed out in the previous slide, I pasted them over here. So one is prothrombin, fibrinogen, I already mentioned, and heparin. So prothrombin, fibrinogen, and heparin, these three plasma proteins are produced by liver and secreted over here. That is released into blood. So you already have come across these terms in your 10th class circulatory system. And there was an important question on blood clotting mechanism over there. So you know about prothrombin, fibrinogen, and heparin. Heparin, I told you yesterday, it helps to keep the fluid, that is blood in our body, in the liquid state, doesn't allow the blood to clot. That is its function. Let's talk about mechanism of blood clotting in short. Whenever we meet with an accident, the tissue gets damaged, you find blood was getting oozed out. That time there, you find blood being blood forming a clot within few minutes. Actually, it should not take more than 2 minutes. Within 2 minutes, maximum 6, people say. Within that time, blood should form a clot. Otherwise, there is continuous flow of blood from the body, which is not good for our body. So that time, the tissues, blood tissues, that is arteries, veins, when they meet with an accident, whenever they are damaged, so the walls secrete, they release platelets and even thrombokinase. Thrombokinase is actually an enzyme. See, 3 things happen whenever formation of the clot has to take place. First thing, vasoconstrictors, they work and they try to, what are vasoconstrictors? Yesterday I taught you about that. Mast cells secrete vasodilators and vasoconstrictors. So vasoconstrictors try to contract muscles of the arteries, they contract so that the flow of the blood is restricted. This is the work of vasoconstrictor. This takes place. Second thing is that platelets are released over here wherever the damage takes place from the damaged tissues, that is artery vascular tissues. Platelets are released, and these platelets, they get stuck to each other, forming a plug-like structure. so that they don't allow the blood to come out. This is another one. And third one is that there is formation of a mesh or a network of structure being formed over there, which will try to trap all the blood cells so that the blood doesn't move out. So that formation of the net that takes place, that is because of one important protein, which is nothing but this fibrinogen. So what is happening over here is that thrombokinase, which is present, this is released when there is damage. When this is released, that time one substance, what we have learned over here, prothrombin, this gets activated. So that enzyme gets activated and it gets converted into thrombin. Then thrombin in turn, again, see, this is a cascade of reactions. So I'm just talking about clotting of the blood in 3 steps, but it is not in three steps. It is 13 steps process. So thrombin gets converted again into, it converts fibrinogen protein into fibrin. Now, dear students, please don't get confused. See, I said that blood doesn't have fibers. That's fine. But there is a protein that is available in blood that is called fibrinogen, and this is soluble. Whenever there is a need for our body, that time, this fibrinogen, it produces fibrin, that is, thread is produced. When there is a need to protect the stop of flow of blood, that time that fibrin is produced under these circumstances when these reactions take place. So this fibrin forms a network like meshwork of structure so that the cells get trapped over there and there is no flow of blood, thus preventing loss of blood. So here is the pictorial representation of what I taught you over here. So whenever there is damage to this blood over here, damage of the blood takes place, you find the blood coming out. Their platelets first are released, vasoconstriction takes place, the blood smooth muscles, they constrict, right? Then the volume becomes small. Second thing is that the platelets are released. This activated platelets, they all come together, they get stuck together, forming a platelet plug. And the third one is that all those 3 reactions take place, and you can see there is fibrin being produced, and that fibrin acts like a mesh so that further loss of blood doesn't take place. So in 3 steps, these things take place. So those are the plasma proteins which help in this way. Now, let's try to understand the importance of the other plasma proteins. What are the other plasma proteins left out over here? Albumins and globulins. Albumins are very small proteins, abundant proteins that are present. What is the main function of albumins and globulins is that they are the transport proteins. They help in transporting those enzymes and hormones. We have studied over here, no? These enzymes and hormones. So these enzymes and hormones are transported with the help of these albumins and globulins. But albumin also has got another important function to do. And what is that function is that it tries to maintain something called as oncotic pressure of blood. It maintains oncotic pressure of blood. What is this oncotic pressure of blood? Before understanding that, let's look at this picture. See, if at all you want water to be pumped into your overhead tank, this is common process which goes on at everyone's house, isn't it? So water which is present down, right, from there, if water has to go up, you're using a pump, because water has to be pumped, taken upstairs against the gravity of the earth, right? But if water has to come down, you are not using any pump. Why? Because simply due to force of gravity, water is coming down, which is because another term for that we use in common language is hydrostatic pressure. So blood that is moving actually over here in blood vessels, right? This blood vessel, blood is having over here, it is moving because of the hydrostatic pressure that is due to gravity. But this pressure of the blood which is moving, it comes across or it encounters one opposing force from within. And that is because of this albumins. Albumin proteins, they try to pull water molecules back. So suppose I'm going straight, right? Hydrostatic pressure, I'm water molecule, I'm going straight. But oncotic protein which is present behind me, it will pull me back. That is called oncotic pressure. So albumins help in maintaining this oncotic pressure. It is also helpful. This pressure is also helpful. Where it is helpful, we'll study when we come across lymphatic system, right? So just see, look at this. Hydrostatic pressure will help the water to come out, whereas over here oncotic pressure helps the water molecules to come back into the blood vessels, because we have studied that plasma consists of 90 to 92% of water. Now talking about pH of blood, blood is actually defined as it is opaque liquid. It is opaque liquid which will not allow slightly opaque, you can't see through blood the other side. So opaque liquid, and it is alkaline in nature. So pH of the blood is around 7.4. Dear students, plasma proteins that we have learned so far about, they also help as or they work as acid base buffers. You know what are acid base buffers? Buffer is any material which will try to maintain the pH of any material constant, whatever it is. Even though you try to add little bit of acid or little bit of base, you try to increase slightly acidic nature or basic nature of a substance, still the pH is maintained because of the substances present in them, which are called as acid base buffers. So plasma proteins act as acid base buffers, maintaining the pH of the blood to be slightly alkaline. You know, on a pH scale, you can rate the acidic or basic nature of the substances. So the scale being running from 1 to 14. So if the pH value of any substance lies from 1 to 7 or 0 to 7, then we call these substances are acidic, and from 7 to 14 it is basic. Exactly, any substance which is lying over here at 7 is neutral, like for example our water. So blood is slightly alkaline in nature with 7.4, very close to neutral point. No, that is why we say slightly alkaline. So if you are talking about hydrochloric acid which is present in our stomach, the word itself says it is acid. That is the reason pH of stomach is 1.8, very much acidic. Now, where is this blood produced? I'm talking about the production of blood. The process of production of the blood is called as hemopoiesis. 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 Formation of the blood is called hemopoiesis. The organs where blood is produced is called as hemopoietic organs. Now, yesterday again, I already taught you that liver and spleen are considered as cradle of RBCs, of blood we say, as well as graveyard of RBCs. Why I'm saying RBCs? Because it is the cells which die over here. That is the reason. So during embryonic development, we find that this yolk sac, which is mesodermal in origin, yolk sac, this actually does the work of production of blood. That means when an embryo is growing, fetus is growing, right, in the womb of a mother, uterus, there you find it is being surrounded by extra 4 membranes. So what are those extra 4 membranes? You have already learned in 10th class: amnion, chorion, allantois, and yolk sac. Generally, in all the other organisms where the eggs are being laid outside the body, there you find yolk sac playing a major role, because it is the one which supplies food to the growing embryo. But in case of mammals, you find the embryo grows within the body of a female. So the growing embryo draws its nourishment, its oxygen, everything supplied from the mother's body. So this yolk sac has no role over there. So yolk sac is simply there, but then it will not do the work of supplying nutrients and all. So there are 4 important extra embryonic layers, one being yolk sac. The work of the yolk sac initially over here is to produce blood. This is one during embryonic and fetal stages. Similarly, you also find liver, and then this is liver. Liver is an angel organ of our body. See, we have seen so many times liver coming in our conversation, in our discussion. And this one is spleen. Yesterday I taught you about spleen, right? Spleen's outer structure we studied, saying that reticular tissue actually protects it. So these are the hemopoietic organs, right? But as you keep growing, then what is the place where now as an adult, right, when we need blood, so which is that part which produces blood? It is this bone marrow, which we have been time and again speaking about this. Actually, it is a red bone marrow. This red bone marrow which is present in the epiphyseal region, this is the one where production of blood takes place. What is the difference between this yellow bone marrow and this is that initially this is also red in color only. But as time passes by, here you find deposition of fat cells, making this marrow as yellow in color. That is the reason this is yellow bone marrow. that is fat is stored over here, and this is red bone marrow where blood is produced. This is only an example of a marrow cavity I'm showing over here. So wherever marrow is there, red bone marrow is there, there production of blood takes place. As I told you earlier, that blood is actually a connective tissue, and it has also got matrix, which is plasma, and then cells. Cells part of blood, we call it as even formed elements, FORMED, formed elements, it is called as. So we have three types of cells present over here: red blood cells, Rbcs, and platelets. So the total percentage of volume of RBCs in blood, in the total blood, 4 or 5 liters of the blood, how much of volume this RBC is actually occupying, if you take its percentage, then that is called as hematocrit value. This is a very important question. What is hematocrit value? The percentage of the total volume occupied by RBC in blood is called as hematocrit value. Now let's study about RBCs. In short, when you look at the points over here, of course, this is a summary of red blood cells. Red blood cells are also called as erythrocytes. They are also called as erythrocytes. Red blood cells, erythrocytes, or another term for this is red blood corpuscles. Red blood corpuscles. So why is this blood red in color? It is because of the presence of one important protein molecule, hemoglobin. This is called as pigment, which is hemoglobin. In the center of this hemoglobin, you find actually globin protein, and on the 4 sides, imagine that it is just like a box, rectangular box, and the 4 sides are there, 4 corners, right? So on the 4 sides, you find 4 chains present. So here you find globin particle, and there are 4 chains present. So those 4 chains are polypeptide chains. Polypeptide, poly, many. Peptides are the bonds created attaching or linking all amino acids. So all the amino acids, they join together to form a chain, which is peptide chain. Polypeptide, many such links are existing. So 2 beta chains are there and 2 alpha chains are there. So to this globin protein, there are 2 chains attached on above and 2 chains down. So 2 are beta chains and 2 are alpha chains. Now here in this 2 chains, you also find presence of heme part. This heme part is nothing but iron, that is ferrous ion. So one ferrous ion takes 1 oxygen molecule. One ferrous ion can carry 1 oxygen molecule. So there are 4 ferrous ions. That means 1 hemoglobin molecule can carry 4 oxygen molecules. So first thing is that erythrocytes, red blood cells, they are red in color because of the presence of pigment, which is called hemoglobin. Spelling is wrong over here, HAE it should be. So because of the presence of hemoglobin, 1 hemoglobin molecule consists of globin part and 4 chains are present. We call 2 of these chains as beta chains and 2 of these chains are called as alpha chains. So in each of these chains, you find one heme part and then you find this polypeptide chain. This heme part, what we are referring to is ferrous ion, and each one carries oxygen. So it forms oxyhemoglobin formation. The clubbing of combining of oxygen with hemoglobin is oxyhemoglobin, which takes place in lungs, and then this hemoglobin gives away oxygen to tissues, which is dissociation. So this is the importance of this hemoglobin. And if you find any kind of destruction taking place with reference to this chain, then people are said to be suffering from disease called as thalassemia. You have also heard this name in your textbook, last year's textbook, 10th class, as well as even on television. Let's talk still about red blood cells, erythrocytes. The shape of the red blood cells in general in mammals, we call this as biconcave in shape. It is biconcave. You know the meaning of the word biconcave? You find over here a depression on both the sides. So it is biconcave in shape. Suppose you consider a spoon. So the depression part of it has got concave shape, right? The other side of it is convex. So shape of erythrocytes, it is biconcave in shape. Biconcave, otherwise you can also have circular shape. But this is different when you talk about camels and llamas. generally it is given in the textbook that camels and llamas, they have elliptical shape RBCs, elliptical shaped RBCs. But even research is going on what kind of really, the shape of the cells is, or another important difference between mammal cell and camel cell is that human beings, we ourselves don't have nucleus. Our RBCs are enucleated, letter enucleated. Enucleated means absence of nucleus. Whereas it is said that camels and llamas, these are camels, of course, these are llamas. Llamas are these are the animals found in South American continent. They are animals which appear like small camels. They are not camels, but another variety. So in these two type of mammals, it is said that there is nucleus present in RBCs, but still research is also going on even in this area, right? So why there is no need of nucleus over here? Why there is no need of nucleus over here? It is because the main purpose of RBC is not to take part in any metabolic activities. It is simply to carry oxygen. So they don't want, these cells are not in need of any nucleus to control them. Their work is only to transport oxygen. That is the reason they got rid of. Initially, the cells will have nucleus, but as they develop, as they become matured, as they become functional, they get rid of all the important organelles. It is mitochondria, nucleus, etc. because they have to carry only oxygen. Okay. Now, where actually the red blood cells formation takes place. Suppose body is in short of oxygen supply. That means oxygen supply becomes less when the cells are less. Which cell is less? RBC is less. So that time, kidneys play an important role. We know that actually kidneys help in filtration of the blood, but that time it is the kidneys which recognize, oxygen is less. That means RBC is less. So kidneys release one enzyme which is called as erythropoietin. This is released in blood. When this is released in blood, this goes to bone marrow, and then bone marrow starts producing more RBC. So it is erythropoietin which plays an important role in the production of RBC whenever body is in short of oxygen or RBCs. So this is the same example again. We can see over here the cells when there is decreased oxygen, kidneys understand that and they release erythropoietin. Then bone marrow secretes it produces lot of RBCs. Now, what is this? We call this, the pronunciation of this is Ruulux over here. Can you see this over here on the top? These are the coins. These are the coins that we use in our daily life. Stacking up of the coins, you can see over here, right? In the same way, you find that hemoglobin molecules, they are also actually found stuck up like sacks. They are also stuck up like stack forms, like the coins. So this is called as Ruulux. This is called as Ruulux effect, we say. This is because of the disc-shaped structure of the hemoglobin that they get stuck up like this. So diameter of RBC is around 7.8 micron, whereas the thickness is about 2.2 micron. Now, formation of the blood, as I told you earlier also, directly in our body, blood is not produced, blood will not get released. I mean, cells will not get released from bone marrow as RBCs together. No. First, you find some cells being released, and those cells have come out. Okay, now they are not differentiated. Cell will not come out and say, I'm RBC. It will, depending on the need of the body, it gets now differentiated into what kind of function it should take up, whether platelets, whether WBCs. The fate is decided afterwards. Now, for example, whatever cells come out from this picture, you remember I said when I was teaching investing time, investing bones, that will come across this. So here you can see that these are the cells which come out from bone marrow, right? These cells, they divide and they produce a lot of cells. The cells which are produced from bone marrow, their fate is not decided here. What do we mean by fate is not decided and they are undifferentiated? Suppose there is a cut on my skin. Then what happens over here after a few days? You find the neighboring cells which are on both the sides, they divide because of mitosis. Number of cells are produced and the gap is filled. That means the dividing cells are all of one type, that is skin cell. It is decided. But if you take over here, the cells coming out from bone, right? We call them as stem cells. They are multipotent cells. They are stem cells. So the fate of this stem cell is not decided. Cells get divided. So many cells are born. Now the cell has the option of becoming a lymphoid stem cell. Lymphoid stem cell means this will afterwards become WBC. There are types of WBCs also. And otherwise it can just come on to this side and become myeloid stem cell. So if myeloid stem cell is there, again, this can become RBC. It can become platelets through a process. See, this stem cell, it becomes one big cell, converts into a big cell, which is called as megakaryocyte. Megakaryocyte, a large cell. So megakaryocyte, it breaks up, it ruptures. One cell ruptures, breaks up. This is called as fragmentation, giving rise to 1,000 platelets. So platelets are produced like that. So myeloid stem cells can become RBC, or myeloid stem cells can give rise to granulocytes. WBC is another variety. So what I want to tell you is that RBCs, WBCs, whatever they are produced in bone marrow, directly they don't come out of the bone as bones, RBCs, WBCs. There is a process which takes place wherein the cells are produced, then they get differentiated. Okay, I should become WBC, I should become RBC. So the fate is decided. The differentiation takes place afterwards, and then the development, maturation, and functional. So that process is only this over here. So under that process only you find proerythroblast, then erythroblast. This becomes in one of the stages reticulocyte. Why I want to talk to you about reticulocyte is that you find actually presence of nucleus still here, and nucleus is given off in RBC here. It gets rid of nucleus over here in this particular stage. Okay, let's talk about the number of, based on the number of RBCs present, we can define conditions, different conditions in the body. Normally, per cubic millimeter of blood, we should have, males should have around 5 million RBCs, and females 4.5 million RBCs. That is a normal condition. If the condition is less, I mean the cells are less in number, then we call that condition as erythrocytopenia, and the person is said to suffer from anemia. We have already learned this term, anemia. We say when somebody looks very pale, very tired, very dull, we say, what, you're an anemic patient? Or the person say, yes, I am suffering from anemia. People suffer from anemia due to various reasons, and one of the important points is deficiency of iron. So anemia causes can be anything. See, for the formation of RBC, you need to have B12 and calcium. These two are very crucial. But people also suffer from anemia. If they don't have iron in their body, then RBC is not produced. So these are all the deficiency causes for anemia. And symptoms, they suffer from all these diseases: tiredness, headache, dizziness, all these. They are not active, right? They suffer from all those things. On the contrary, if the number of the RBC is more than that 5 million per cubic millimeter, then what happens over here? The condition is called as polycythemia. Polycythemia over here means there is increased RBCs. What happens if increased RBCs? We can say, yes, it can carry more oxygen. No, that is not the case. What happens if the number of the RBCs is more? The movement of the blood flow, it decreases. And slowly you find clots being formed everywhere in the blood vessels. That is nothing but one of the cancers, blood cancers. So that was all about RBCs, red blood cells. So dear students, we have completed today so far about bone. We have started with the blood. We understood plasma. We understood even RBCs, one of the cells in plasma, in blood. Tomorrow, let's continue with WBCs, platelets, lymphatic system, and the other parts. Thank you for being with me today. Vijayalakshmi ma'am.
Yeah, but thank you very much, madam. It was very detailedly explained. Children, please revisit the channel and complete the running notes of the webinars. And please stay tuned, because we'll take just 2 minutes break and we'll come back with English session. Thank you, children. Thank you, ma'am.
Thank you, ma'am. Thank you.